Okay, we're going to talk about the synapse. And of course, that's where the gap between two nerve cells. So if you imagine this black nerve cell on the left, and there's an action potential which is moving down there, a wave of depolarization. Remember, as sodium ions are entering the cell, and the membrane potential goes from the inside of minus 70 millivolts, becomes more positive as positive sodium ions enter, going up to plus about 55 millivolts. And when it gets to the end here, the synapse, it can't do anything because you haven't got any cytoplasm in between these two cells. There's a gap. So how does the message get across? Well, the message gets across by a small molecule called a neurotransmitter. Okay, and the neurotransmitter, there are several different ones, but we're going to talk about acetylcholine, which is one of the best characterized neurotransmitters. And that's a small molecule which diffuses across that gap and triggers an action potential in this second cell here. So you get another action potential in there, and that will then move along. Okay, so let's see in more detail how that's done. Okay, so what I've drawn here is we have got on black, they would call it, that's the black cell, that's the presynaptic membrane, and on the right, the blue, the postsynaptic membrane. Now, what these things here are called vesicles, okay, the membrane-bound vesicles, and they contain the acetylcholine, which is the neurotransmitter. And I've drawn that as little red dots. So these diagrams are, of course, not to scale. We've got lots of molecules of acetylcholine in those vesicles. And somehow, one of those vesicles has got to go up to the membrane and fuse, and then it will disgorge its contents of acetylcholine molecules into the gap, and we'll talk about what they do in a minute. But let's see how the steps leading up to that. So these little red channels here, these are supposed to be sodium voltage gated. So as the wave of depolarization passes down the cell, you're going to get sodium ions are going to flood into there, and that will depolarize it, go to plus 50, plus 50 millivolts or whatever, and be negative, and that will, the wave of depolarization will continue, get sodium ions coming in here. And then these little green ones, these aren't sodium gated, voltage gated channels, these are calcium voltage gated channels. And when the membrane potential increases up to 50, when you get depolarization, calcium ions are going to flood into the cell because the extracellular calcium concentration is a lot higher than that inside the cell. The calcium concentration inside cells is very, very low. Okay, now on these vesicles, you have some proteins which bind calcium, and when they bind calcium, that causes them to do what's happening here, them to fuse with the membrane and release the acetylcholine molecules here. Interestingly, you may have heard of Botox, which is causes paralysis of muscles. So it's injected into people's foreheads to paralyze the face muscles and make you look less wrinkly. Botox is actually produced by a microorganism. It's a toxin, and what it does is it messes up the calcium binding proteins, and it stops those vesicles from fusing, and so it stops the acetylcholine being released, and so the nerve impulse doesn't go anywhere, can't go, it stops, and so whether you don't get stimulation of a muscle, whatever, so you get paralysis. Anyway, let's talk about the acetylcholine molecules. They are released. Then on this side, on the postsynaptic membrane, these black things here, these are the acetylcholine receptors. And what they are is they are sodium channels. They're not voltage gated. These are sometimes called ligand gated. The ligand being the acetylcholine. And when the ligand, the acetylcholine, binds to the receptor there, I draw it there, that changes the shape of this transmembrane protein and allows sodium ions to pass into the cell. Okay, so there's acetylcholine there. And of course, when sodium ions pass into the cell, that will change the membrane potential to plus 50 millivolts, whatever. It will depolarize it. So you've caused, and that will then stimulate an action potential in this second cell. Okay, so that's the basic process of how acetylcholine, one neurotransmitter, enables the message to be transferred across the synapse. Let's just talk about acetylcholine itself in a little bit more detail. Okay, so if you're interested in the chemistry of it, you can see we've got acetylcholine here. Acetylcholine is an ester. There's the ester linkage. And you've also got a quaternary amine there. Now, in the synapse, this acetylcholine is broken down into, this is choline here, and ethanoic acid. And you've broken the ester bond, so it's an ester, it's breaking down an ester, and the enzyme that does that is called acetylcholine esterase, and that is absolutely essential that that's there, because if you don't break this down acetylcholine, it's going to remain in the synapse and it will keep firing the nerve repeatedly, and that's not what should happen. You just want the message to go across. So very rapidly, as soon as the acetylcholine is actually bound to its receptor, it will be hydrolyzed by the enzyme acetylcholine esterase and broken down, and it will stop stimulating it. And interestingly, you may have heard of the Salisbury poisoning, which the substance Novichok. Novichok is an inhibitor of that enzyme, so it stopped that working. So continually the nerves are being fired. Acetylcholine is important in the whole of the nervous system, and so it causes paralysis and death, and I think some people survived, but then they had a lot of mental health problems, which is not surprising really when you think that it is going to mess up the entire nervous system. Okay, let's just talk about, so that's the mechanics of what happens at the synapse, but if we just talk about the importance of synapses generally in the nervous system just for a minute before we finish. What I've drawn here, this is a neuromuscular junction, it's supposed to be. So the black thing is the neuron, and the little red thing there, that's the muscle cell. The way the muscle contracts is, well, you have a synapse between the nerve and the muscle cell, and you have acetylcholine receptors on the muscle cell, and that causes depolarization in the muscle cell, it causes contraction. So not only is it so important between nerve to nerve connections, but it's also nerve muscle connections. What I've tried to draw here in this one here is a nerve cell, that black nerve cell, and there's 3 synapses, okay? There's 3 different synapses there with 3 different nerve cells, 1, 2, and 3. And this is massively oversimplified because most nerves in the brain are going to have literally thousands of different synapses connected to different lots of different cells. Now the synapses they're summative. So imagine synapse number 1, that releases some acetylcholine, and you get a little bit of sodium going into the cell there. But that in itself it's not going to reach the threshold potential, or is it? It's not going to be able to minus 50 or whatever. It's not going to be able to stimulate the nerve cell. But if 2 and 3 also release acetylcholine at that time, that will lead to a greater influence of sodium coming in. So that may be sufficient to trigger a nerve impulse there. So the interaction between lots of different nerve cells, I've only drawn 3 interacting on 1, that's how the brain is able to carry out such complex functions. Synapses are the basic unit of the brain, really, and the way the brain works. Finally, there are other neurotransmitters. Some of them you may have heard of, and these tend to be important in the brain, as well as in the peripheral nervous system. So you may have heard of serotonin, which is made from the amino acid tryptophan. That's very important in maintaining mood. There's one, GABA, gamma amino butyric acid. That's pretty important. Glycine, the amino acid glycine, also doubles up as a neurotransmitter, and glutamate, another amino acid, also doubles up as a neurotransmitter. So these are also pretty important, but the basic principles of how the message is transferred across the synapses is common to all of them.